Alright, so I wrote this down on the board, but don't feel like you have to memorize all of it because it looks really overwhelming. But we're going to be talking about each part of this, and this is called cellular respiration. I'm sure you guys have learned this in biology, right? Do you guys remember? So everyone's heard of glycolysis, citric acid cycle, or TCA cycle, Krebs cycle, yeah. Electron transport chain, yes. Okay, cool. So like I said, don't feel like you have to memorize like the specific parts of it, but while we're going through this, you may get a little lost once we're talking about anything more specific. So always look back at this if you feel a little confused. And also feel free to stop me. I tried to summarize it as best as I could because the whole point of all of this is to know how ATP is being produced. And ATP is used throughout our whole bodies and for like different cellular processes, and today we're going to talk about like cell potential or charge equilibrium in cells. That has to do a lot with action potentials, and then muscle contraction of the heart. Okay. So ATP. So ATP stands for adenosine triphosphate, and this is the storage and transfer of chemical energy within the cell. So this is ATP right here. And so you'll notice that there's an adenosine, which is actually nitrogen based. Do you guys remember ATGC in nitrogen? Yeah. So the A is from adenosine, and then it has 3 phosphate molecules. And the reason why we're talking about this is because whenever ATP is hydrolyzed, that means that one of the phosphate groups is being broken off from the ATP. So whenever that phosphate group breaks off, you get energy for cells. So this is how that energy is being delivered. These, like the entire ATP molecule is really unstable, so it really wants to get rid of one of those phosphate groups. So that's why it's really perfect for our body to have ATP. It's really easily hydrolyzed, which means what? What did I say hydrolyzed means? Yeah. And the way that we generate ATP again is energy from food. So we have, after the phosphate group breaks from the ATP, we get ADP, which is adenosine diphosphate. So triphosphate, 3 phosphates, diphosphate, 2 phosphates. So that's why it's called ADP. So once we get that energy from our food, one of the phosphate groups is being added back to the ADP to make ATP. Okay, so does that make sense? Mm-hmm. All right. And so this is how ATP is generated. So we have glycolysis here. I'm going to just quickly summarize this. Glycolysis here, then we have the electron or Krebs cycle, and then the electron transport chain. So glycolysis happens in the cytoplasm, and the Krebs cycle, electron transport chain, and oxidative phosphorylation it's just like a fancy term for saying that ATP is being produced within the electron transport chain. Happens in the mitochondria. So that's why it's called the powerhouse of the cell. And so, like you notice here, ATP is being produced after glycolysis, after the Krebs cycle, and after the electron transport chain. But just know that a mass amount of ATP is going to be produced in the electron transport chain. So do you guys remember talking about anaerobic, aerobic respiration? Mm-hmm. What do you guys remember about that?
Anaerobic more ATP than anaerobic.
Yeah. So like she said, so whenever there's oxygen present in the cell, we're going to go through this entire process here: glycolysis, Krebs, electron transport chain, and it creates more ATP. About 30. You don't have to know that. It's just so you can see that it's a lot more than an anaerobic respiration, which is only 2 ATP. And with anaerobic respiration, what else do you know about that? What is the product of anaerobic respiration?
Lactic acid.
Okay, where's lactic acid? How does that apply to us?
It gets in your muscles.
Okay, good. What about, and then alcohol production in other organisms, like especially if they don't have a mitochondria, because what types of cells don't have mitochondria within other animals? This may be a little extra. Like bacteria don't have mitochondria. Yeah, okay. So yeah, just the point of this is that aerobic respiration makes more ATP. So I know this looks really overwhelming. You don't have to know that. I just want you to see that in glycolysis, it's a really long process to breaking down glucose into 2-pyruvate. But what I do want you to focus on is the products. So we have 2 ATP molecules that are produced after glycolysis. 2 NADH, which are electron carriers. Have you guys heard NADH, FADH2 in biology before? All you have to know is that they carry electrons, and that electron is that hydrogen. So with NADH, once it gives off an electron, it becomes NAD plus because that H is being removed. And then 2-pyruvate, which is at the very end of glycolysis. And that pyruvate eventually is going to turn into acetyl-CoA. You don't really have to know this, but it goes through this cycle of breaking carbon bonds, and eventually you'll end up with more electron carriers, carbon dioxide, that's the carbon dioxide we produce, and then 1 ATP. And so I'm talking about these electron carriers because this is really important for the electron transport chain, or oxidative phosphorylation, which I said means that ATP is being produced in the electron transport chain. So I know I went through that a little fast, but now we're, or after the citric acid cycle, which is in the mitochondria, now we're in the inner membrane space of the mitochondria. So this is where the oxidative phosphorylation occurs, where the mass amount of ATP is being produced. So those electron carriers eventually, there's like a series of reactions that happen within here, and eventually it pumps protons out of the cell. And these protons are really important for the production of ATP because do you guys know what this molecule or this protein is right here?
No.
What does it produce? ATP, right? So what do you think is the name of this? ATP synthase, yeah. So this is the huge protein, the most important protein for ATP production. So with those series of reactions of the electron carriers,
what's it called again?
ATP synthase. Those protons that are being pumped out will be pumped into the ATP synthase, and eventually will have enough energy to add a phosphate group to ADP, which is what? Adenosine diphosphate, to add a phosphate group to make it adenosine triphosphate, and that's energy, right? And so then this ATP is going to be distributed throughout different parts of the cell, throughout our body, to carry out different reactions that require energy. Okay, does that make sense? So you guys have any questions before we move on? No? So can you guys, without looking, what's like the first step of cellular respiration? Glycolysis. After glycolysis is, if there's oxygen present, Krebs, and then after Krebs, electron transport chain, oxidative phosphorylation. And then we have the production of ATP. Yeah? Eventually, you'll have to, because I spent like an entire 2 weeks when I was taking biochemistry learning like these reactions, but just don't feel overwhelmed. Don't try to memorize all of this because I still get confused when I try to think about it. Because like I said, it was like an entire 2 weeks of learning how like that worked. But just know that all of this is for the production of ATP. So do you guys remember passive versus active transport in biology? Yeah. So what's passive transport? It doesn't require energy. Yeah, so it doesn't require energy to move a molecule across the cell membrane. What about active transport? Yeah. So active, think you need energy. And so for diffusion, can someone explain what's happening over here with diffusion?
Aren't the molecules moving from high gradient to low gradient?
Yeah, so there's a high concentration of a certain molecule here. We don't know what it is. And it's going to move along with the gradient, which means that it's going to want to be at equilibrium. So we're going to want the same number of molecules on the outside versus the inside. And so that happens passively without any energy. facilitated diffusion. Do you guys know what facilitated diffusion
It
is?
requires something to start doing that, like a
Well, think of
channel.
what does facilitated mean?
It gets carried easier.
It gets help. So it doesn't require energy, but it requires a protein to be able to move that molecule across the cell. And so that's why it's called facilitated diffusion. This still doesn't require energy, because we're moving from a high concentration to a low concentration inside the cell. And so now the most important part, which is where we're going to talk about the sodium-potassium pump, is an active transport protein, which is, it could be either a pump or like the ATP synthase. It still requires energy, right? So this is active transport, because what do you guys notice here? We're trying to get these two molecules to go inside the cell. Why is that a problem, do you think? Or not a problem, but why do you think we can't do that without energy?
Because it's probably charged.
Because we're trying to be at equilibrium, and if we add one more of these in there, will it be at equilibrium?
No.
Will there be the same number of molecules inside and outside?
No.
No, right, obviously. So we have to add energy to be able to move that inside. And so this is an example of the sodium-potassium pump, which is where y'all left off, right? The sodium-potassium pump. Okay.
I guess a question.
Yeah.
Can you go back and, you said gets help with, requires a protein. Can you talk about that a little bit?
Yeah, so with diffusion, for example, it doesn't need a protein across the membrane. It's either small enough, or I think one of the requirements is like it's like non-polar. Do you guys remember what that is? Yeah, I know this is going back to biology, but pretty much this doesn't require a protein. With facilitated diffusion, what the protein or the molecule may be too large to pass the membrane, so it needs a protein to carry it over. Or sometimes the charge of it, since I don't know if you guys remember, but on the inside it's negative and on the outside it's positive. So sometimes we have a molecule that's either too, it's negative, positive. It's too negative, and negative charges collide, so it still needs a protein that will help it go through. I don't know if that makes sense.
Can you give an example?
Yeah, so like sodium channels. So okay, so if you have the membrane, these are the phosphate heads, and then these are the fatty acid tails. We said that on the inside it's negative, right? And the phosphate heads are positive. So let's say that we have a negative molecule that's trying to get in. It can pass through, or it cannot pass through without a protein, because if it tries to, hold on, let me try to think about it. If it tries to pass through the membrane when it's negative here, it's not going to want to go in because it's going to repel it. So then we use a protein that has a positive inside, so that the negative will be attracted to it and go through. Does that make sense? Okay.
I mean, do you have an example of a protein, or is that just a type of protein?
Well, there's like the potassium channels and the sodium channels, because they use the sodium channel and the sodium uses the sodium channel to go through.
But there's not a name for that protein. It's just a positive protein.
Sodium channel protein,
Okay.
potassium channel protein, calcium channels.
Yeah, that makes sense.
Then there's one with glucose, the glucose transporter. Those are just the ones I can think of from the top of my head. Okay. So the active transporter would be the sodium-potassium pump.
I hate to go back to this,
Yeah, no, it's fine.
but the glucose transporter is that's the protein you're talking about for like glucose. It has to have that transporter we talked about that briefly at one time, so that glucose transporter is a protein.
Yeah, it's a protein on the membrane that helps glucose pass the cell membrane.
That makes more sense. Okay.
Yeah, because there's like these phosphate heads are really polar. Like one side of it's really positive, the other side of the tail is really negative, and some molecules, depending on their charge, can't pass through. It needs something to help it pass the membrane. So yeah. So now when we're talking about the sodium-potassium pump. So I don't know if you guys remember when we were talking about the study strategies, when I said salty banana. Yeah, okay, so this is really important when we're talking about the cell potential, which means like the charge of inside of the cell. And so I'm just gonna write it right here. So do you guys remember what I said about salty banana? What does that mean? So this is a cell, that's the membrane. So where's the sodium going to be if this is a salty banana? On the outside. So sodium on the outside of the cell. And then bananas have potassium, so a lot of potassium is going to be inside of the cell. Okay, so what the sodium-potassium pump does is that it makes sure that the electrical potential of the cell, so the charge of inside the cell, remains, I think it's like around negative 70 millivolts. Yeah. So that just means that the inside of the cell is going to be negative and the outside of the cell is going to be more positive. And so when we have the sodium-potassium pump, it makes sure that potassium goes in and sodium leaves the cell. Okay? And so this is how it makes it at equilibrium whenever the cell's potential changes, and let's say that it becomes more positive, the sodium-potassium pump will want to bring it back to that negative, so that's what that does. And with the number of sodiums that go in or that go out, it's 3, and 2 potassiums go inside. So the way that I remember it is, my name, I know this may not apply to you, but my name is like Karina, it's a K. I don't have a middle name, so I only have like Karina Gomez. So 2 go in, and then sodium 3 out. I don't know how I remembered sodium, but that's the way I remembered it. But yes, I don't know how else you guys would. I don't know, do you guys have an idea how you can remember that 3 sodium go out and 2 potassium in?
You know the Minion song? It's like the banana song.
Okay,
it says na 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 more than banana itself. So
okay, so you can say na na na. So 3 sodium go out of the cell, and then the opposite, 2 potassium go inside the cell. What else? Yeah, so like we said, outside of the cell is what charge? Positive or negative? Positive. Inside the cell, negative. And I want to show you guys this video, that way, because I think it's pretty helpful when we're talking about this, so give me a sec.
Oh, and then potassium, you know when you're mad? When you're salty? Hey, I have a lot of free time.
Active transport is a energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites with 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
Alright, so did that make more sense? Alright, so now with passive transport, because we also use passive transport for the wow, I completely forgot. The action potential, sorry. We have the sodium channels, calcium channels, potassium channels, and chlorine channels. And so with these, like we said, it doesn't require any energy, so it's going to want to flow to the lower concentration. So if we have a high number of sodium on the outside versus the inside, it's going to want to passively flow in. Same thing with calcium, same thing with potassium. So whenever these are involved within the cell, whenever they're activated, it's going to cause a shift in the charge inside of the cell. So if, let's say, sodium rushes into the negative cell, sodium is positive, right? What would happen to the charge inside of the cell? It would get more positive, right? What if potassium leaves the cell?
That's more negative.
It gets more negative because that positive, the positive is going to leave the cell, leaving it more negative. Yeah. And so that's when we start talking about action potentials. So just know that there are, we're going to talk about the nerve cells and cardiac myocytes, which are the muscles in the heart. And so with the action potential of a nerve, which is what we're going to talk about first, it happens really fast.
And a quick question.
Yeah.
Is this where they refer to as the, like you have the pumps, but you have channels that leak?
Mm-hmm.
Is this what they're referring to here, this passive part where they're leaking?
Yeah, it's passive. It doesn't require any energy.
Okay.
The only thing that requires energy is the sodium-potassium pump, because we're trying to put a lot of potassium inside and a lot of sodium outside. We don't want sodium in here, so it's moving against that gradient. So it's not going to be equally distributed. That's why we need that energy. But whenever like sodium and potassium channels are activated, it's going to want to move from the lower concentration to, or from the higher concentration to the lower concentration to make it more equal. Does that make sense? Okay, cool. So with the terms involved with action potentials, it can get a little bit confusing. So I decided to write them down. Polarization, I talked about this in the beginning. It just means that there is a difference in charge on the inside versus the outside. So like we said, negative inside the cell, positive outside. That's what polarizing means. Depolarization is a shift in that chemical charge where the charge distribution resulting in a more positive charge inside when because normally it's negative inside. So this means that the cell potential becomes more positive. Repolarization is like restoration of the cell potential. So we go back to that negative potential. And then hyperpolarization means that there's another shift in electrical charge where it would make the cell membrane a lot more negative than usual because a normal charge inside the cell is negative 70. So with hyperpolarization, it's like negative 80, negative 90, depending on how much of that charge is getting in. So I would, because I'm going to talk about like depolarization, repolarization, hyperpolarization. So I want you guys to make sure that you have these down before I start talking about the action potentials. So does that make sense, this part right here? Okay, and if you get confused, then I would go back and look at that so that you know what I'm talking about when I'm going over the action potential. So action potential in neurons. So when you think of action potentials, what does that mean to you? Because I know it's just like a word and you know it involves charges, but what does it actually mean? If you think about it more generally.
Like the ability to do something.
Okay, what else?
Like a charge moving down
The charge moving down the neuron, but what's the whole point of moving a charge down the neuron? Yeah, so that's how signal is being sent from neuron to neuron. So this is an entire neuron cell. This is one cell. And so let's say this is where the signal begins, and let's say it's for, I don't know, moving your arm or your, just any type of signal. We're going to get like something in the environment out here that's going to trigger a response in the cell body. Do you guys remember the anatomy of neurons? This is the nucleus.
Dendrites, nucleus.
Yeah, dendrites, the soma, and that's the nucleus. So we get that signal, and once we get that signal, this is where the action potential begins. And each one of these little arrows is like an action potential. And so there's going to be a change in the charge. So normally in the axon, it's negative because it's a cell, right? So it's negative on the inside, more positive on the outside. The way that the signal is being propagated throughout is that sodium rushes in, making it more positive, and then the outside becomes more negative. And so that's going to happen along the axon until it gets to the next neuron. And then that's how the signal transfers from neuron to neuron. But I'm going to talk about more about the changes in charge, but I just want you guys to know that that's what action potentials are for. It's to send that message along. And so the resting potential is whenever the axon is at rest. So it's negative inside, positive outside. And that's the due to the job of the sodium-potassium pump that's keeping the sodium out and potassium in. Action potential is when the signal is being propagated along the axon. So, like I said, more positive on the inside, more negative on the outside. And so this is the resting, yeah.
When you say something causes that, is that the neurotransmitters?
Neurotransmitters, yeah. Or like if we're talking about the heart, like the SV node is it causes its own signals or triggers. So, but in this case, yeah, it could be like a neurotransmitter. Well, it is a neurotransmitter for a neuron, but yeah. So yeah, resting memory potential is negative 70. Sodium flows into the cell, making it more positive. So if you see here, this is the voltage, millivolts, this is just the charge. So it becomes more positive. Once it reaches let's start over. So resting potential, and then sodium starts rushing in. When it hits this threshold, it can't go back. So it's going to shoot up to the action potential, and then this is where potassium ions start flowing out, because we're trying to bring that charge back negative. Okay, so sodium rushes in, makes it positive. It's way too positive in here, so now the only thing that's left is potassium leaving the cell to bring the charge back down. Okay, but eventually it becomes way too negative in here. That's where the hyperpolarization occurs and the refractory period. And so it becomes so negative that now the sodium-potassium pump has to work to be able to bring it back to that resting potential, which is potassium inside, sodium outside. Does that make sense? A little bit. Okay, so let's just real quick. So so resting memory potential, potassium inside, sodium outside, right? What happens here?
Sodium goes in.
Yeah, so sodium channels bring sodium back in. It reaches the top, but it's way too positive now. So now what happens?
So now it's
potassium goes out to make it more negative, but eventually it becomes way too negative. So then now it's the sodium potassium's pump job to bring it back to that resting potential. Okay. This refractory period just means that it's way too negative, so it's going to take a lot more sodium to be able to bring it to cause another action potential to happen, which makes sense, right? It's a lot more negative than here. So we need a lot more sodium to bring it back to resting potential and cause another action potential. Any questions? Okay, so sodium flows in, potassium goes out, and then sodium potassium pump starts working here. Okay, so now before we talk about action potentials and cardiac myocytes, I want you guys to memorize this. We need calcium for heart contraction, okay? So the components of muscles is with myosin and actin, and these are filaments. So the myosin, thick filament. Actin is the thin filament. The way that I remember it is like myosin, I think muscles are thick. Actin, I don't really think of muscles when I hear the word actin. Well, before I didn't really think about muscles when I hear the word actin, and so I think thin. So muscle, thick, actin, thin.
Someone that's active is thin.
Okay, someone that's active is thin, I guess, if that helps you remember. So with muscle contraction, these two filaments are going to slide against each other to create contractions. And like you can see here, this is a cardiac muscle, so we have the thin filaments here, thick filaments here. So you can see that when it slides against each other, it causes that contraction. When it relaxes, they get spread out. This, this is a thin filament and then a thick filament. It's literally thicker than the thin filament. So myosin, thick, actin, thin. Okay, so now this is where we're really going to have to really focus because it is a little confusing. I didn't learn this until I was in biochemistry in college, but I think it's a really good idea for you guys to know how muscle contracts more on like the molecular side of things. So we really have to be aware of the structures that are involved here. So this is a thin filament, which I said was what? Actin. Thick filament, myosin. So myosin has these myosin heads, which is what attaches to the thin filament to be able to move or attach to it and then contract and relax. And so the myosin head attaches to the actin at a specific site, but what happens is that this pink little filament here is called tropomyosin. That blocks the site of the myosin head to the actin. That's why we have troponin, which whenever calcium binds to troponin, it's going to cause this tropomyosin to move away from where the myosin head would attach, and it would cause the myosin to be able to attach again. So let me say that again. Calcium binds to troponin because tropomyosin is blocking actin from binding to it. When calcium binds to troponin, this will move so that the myosin head can attach. Okay. So like I said here, troponin has a calcium binding site, and then tropomyosin blocks myosin head from attaching to actin. So that would cause no contraction. That's all I want you to know right now. Does that make sense? Yes. So let's say there's no calcium. Can the myosin head bind to actin? No. What happens when calcium binds to troponin? It moves, and then the actin head can attach. Okay. So that's why I said that calcium is really important for muscle contraction, because that's the only way that the myosin head can even attach to the actin to cause the contraction. So then, like I said here, calcium binds. It literally moves away from the active site, and then the myosin can attach. Okay, so the calcium is released by the sarcoplasmic reticulum, which is what?
The endoplasmic reticulum in muscle cells.
Yeah.
I knew you were gonna say that.
Okay, yeah. So yeah, the sarcoplasmic reticulum is like the ER in like a regular cell, but or like a basic like cell. But in muscle cells, it's called the sarcoplasmic reticulum, and so that releases calcium. And that calcium binds to troponin, causes myosin to move, exposure to the actin, like we talked about. Okay, so now this is how the myosin actually binds to the actin and the steps involved. So, I don't know if you, yeah.
Is it a big deal that SR it releases the calcium but also reabsorbs the calcium?
Yes, it reabsorbs the calcium.
Okay. So we don't really use it, delete it. It just reabsorbs and use it again.
Yeah.
Okay.
Wait, can you repeat that?
Maybe. The SR releases the calcium, but it reabsorbs the calcium. So that we don't delete the calcium, we just reuse it. Up to some point, then we'd have to reproduce it, correct?
Yes.
Sort of, in theory.
Yes.
You want me to play that video?
Well, yeah, I want to talk about it real quick. Okay, so the first step so this is the myosin head, this is actin, is that the myosin head is going to have the ATP molecule attached to it, which is energy, right? As soon as it hydrolyzes, which means phosphate group leaves to make it ADP, the myosin head, they call it cocking, so it will move up, and it's ready to attach to the actin. As soon as the phosphate actually leaves from the ADP, the myosin head will attach to the actin. And then when the ADP molecule leaves, it will cause the myosin head to pivot, and then the muscle will contract to the left. So it goes like, I have to use my hand in order to like understand it, but it's like ATP, and then it hydrolyzes, moves up, the phosphate leaves, attaches to myosin, and then the ADP leaves, and then it moves. And so then the muscle's moving that way. Okay? So yeah, now I can play the video.
Is it supposed to have some sound?
I couldn't find the original video, so I don't know if this one has sound or not, but this will still show you how it moves, if you still need that picture. An organic phosphate is that extra phosphate molecule. and then it pivots. Yeah, that wasn't really a good video. Sorry. I couldn't find the original. Okay, so does that make sense? How the actin and myosin interact? Okay.
Can you show the thing with your elbow again?
Yeah. So it has ATP. The ATP hydrolyzes, moves up. The phosphate molecule leaves, attaches to the actin, and then the ADP leaves and it moves this way, and the muscle moves that way. Yeah. Okay. I still have to think about it because I don't have this completely memorized. It took me a good while to be able to memorize, okay, ATP, then it does this and that. But I guess the whole point of this is just know that ATP works. We need energy in order to move these cells, and this is for both skeletal, smooth, and cardiac muscles. Okay, so now this is where we start talking about cardiac myocytes, so action potentials in the heart muscle. And so automaticity means that the heart muscle cells can fire on its own without a signal. And so we don't need that neurotransmitters or anything to really cause an action potential to occur. Like the SV node is where the cells can cause action potentials on its own. So we're at resting potential, which is about 90 millivolts, around there, in heart cells. And then sodium enters the cell, the same as in nerve cells, enters, becomes more positive, and this entire process here is phase 0. Phase 1 is when it gets to the top of the action potential, the most positive, and then this is where calcium enters. And so as soon as calcium enters, we have this plateau, and the reason for that plateau is because when calcium is going into the cell, potassium is also leaving at the same rate. And so there isn't really a huge change in the charge of the inside of the cell versus the outside, because we're having positive come inside and potassium leaving. And then repolarization, which is going back to that negative potential. The potassium leaves the cell and it comes back to positive in phase 4. So the important ions involved in this, or cations, is sodium, potassium, calcium. And like we said, calcium is really important for muscle contraction, so that's why we see that in cardiac myocytes and not in nerves. So this is where I would leave it to Randy. Is it, yeah. So does that make sense? Mm-hmm. A lot more sense? Okay, mm-hmm. So what is this called whenever the potential becomes more positive? Yeah, it's phase 0, but in general, what is that term where it goes from a negative to a positive?
It's depolarization.
Depolarization, when it comes down, is repolarization. When it becomes too negative, like in nerve cells, hyperpolarization. And then this is just polarization. Okay, so polarization, depolarization, repolarization, hyperpolarization, repolarization. Any questions about any of this?
You done? Okay, let's go through a couple things right quick. And nothing to add there. I couldn't add anything, but let me go back to and whoops. Look at this. Because we talked about yesterday in my failure attempt to explain this, we talked about this here when we talked about action potentials and electrotonic potentials. Remember the short video on that? Electrotonic potential would be this nerve here. It's very fast and it's good for a short amount of time, but it's not good over distance. So we couldn't have an electrotonic potential in cardiac because it doesn't do good over distances. It only does good over a short amount of time, like me picking up the remote or something, or doing quick nerve muscle contraction, okay? So not good for the heart, because remember, the heart pumps. When the heart contracts, it contracts over a distance, and this will make more sense, especially when we talk about bundle branch box later in EKG interpretation, because it's over a period of time. If you looked at the conduction part of the heart, after the bundle hits, it breaks off into the bundle branches, which comes down, remember that? And then it goes to the Purkinje fibers. If we had an electrotonic potential, it wouldn't make it that far, to say it simply, okay? So we need this action potential here in the cardiac myocyte, or the cardiac cell. So let me flip back over here to this. Whoops, one too far. So these different, you will need to know these different phases and what takes place in these different phases. But as we go forward in here, we look at the action potential of this cardiac cell in this calcium influx here. Sort of makes this longer, okay? It lengthens out that signal, okay? And it's good for heart contraction. It's the sort of the easy way to say it for me. Okay, so you have the depolarization and then the plateau, because the heart contracts over a period of time. I've got this really cool video that I took a video of off of Facebook that I'll show you the heart contracting, and you can actually sort of see it go in sort of a flow. It starts and it sort of runs very quickly across the chambers of the heart, okay? And that's for a later day. But without this, we're not going to get that length of contraction. It's going to be more like a nerve cell, correct? Where it's going to contract very fast and not over time. So the action potential is good for time. It takes more energy to do this, but it's good. It dissipates over time. And that's what we want in the cardiac cell. And again, we'll get back into this on another day, but I wanted you to sort of look at that and think about the way that the heart muscle contracts and over time, and think about because you have to add a number of things to that. You have to add, the electrical conduction system. So when you start visualizing these different things, you say, oh yeah, okay, I can sort of see that as the signals, those, because in the cardiac cell, or the heart, you have three types of cells, right? You have a pacemaker cell, which is at the SA node, right? So the SA node is a bunch of pacemaker cells. Then you have conducting cells, which are going to conduct this, okay? And then you have contractile cells. And that's what we're getting into next, is those three different types of cells, right? But here, and you can tell them, if you were to dissect the heart, you can tell these pathways because they're a different color, okay? They lack actin and myosin, right? These cells don't. Pacemaker cells and conducting cells, they don't have actin and myosin because they don't contract, right? Only contractile cells have the actin and myosin, so they would contract. But I think that's for another day. Let me get my notes from my failure yesterday.
I said SA node, I meant SA node.
Oh yeah, SA node.
I like that.
We just used the word electrogenic response. That's the response that changes this electrical potential. So just a vocabulary word for you. Electrogenic is a change in electrical potential, right? And to make this a little bit more confusing for you is because when we look at this, we have to, the mind, I think, this is opinion, but the mind sort of has to have a picture of something that, like this. It has to have. I can picture that, right? It's a nice picture, and I can picture this sodium and potassium leaving through this little thing here, okay? And I can remember that as a picture, but part of this, while we're learning it, is that you guys need to understand that it gets harder, okay? It gets really difficult in the areas that you're going to go. How many of you guys want to be doctors? Yeah. Welcome to biochem. A taste. I mean, it's not even close to it, but it's just a little taste. You know that it's the taste of when you get that big scoop of bluebell and you put it in here, and you still have some on the scoop, right? It's not all of it by anymore. You got this big, huge bowl of ice cream, but you have some bluebell left on the scoop. What do? You don't wash that off, do you? Oh no. No. You lick it. You lick it off. You go, oh yeah, that's it. This is what that is. This is just that taste to get you going and understand that it gets hard. Not to discourage you, but only to encourage you. Say, hey, I've got to really step up when it talks about from our chemistries, right? General chemistry. It's harder than what you experience now. Organic, sort of now the new doctor stopper, and then biochem is again it's termed the doctor stopper. Okay, but we want to overcome that, which you can. You just have to be prepared to work hard. Okay. But to make it more confusing now, because we have this picture, and we go, oh yeah, I can see that. I can see that picture. But these are, if you remember in the video, this is just this is a cloud. Would that be a good way to describe it? Because that's the way they used it in the video. This is a protein cloud. It's just a cloud. Yesterday in my fatal attempt to describe it, I described it like a cloud of gnats. In the summertime, North Texas, you get these big, huge clouds of gnats, okay? And that's what this essentially is this cloud of gnats. And it's just a protein cloud.
Proton or
Proton.
Oh.
Proton cloud. It's just a cloud.
Oh,
It's not a solid, it's not like a pump, like you would think of a solid, something that pumps, right? And you know that. But it's a cloud, and these things combine with things in this cloud. Okay, you probably don't want to think about it that way because it's too confusing. Okay, but you have to understand that these are things that are floating in the bloodstream, and we have all these receptors and all these different things. So it's just this cloud in these chemicals. When we flip back to here, and I'm not going to go into this because it's not necessary, but I do want you to understand that there's more. There's always more. At each level, where you're going in medicine, you take what it is at that level, right? You guys who want to be doctors, you're taking it at the biochem level, right? Nurses are taking it at a different level. Paramedics, we take stuff at a different level. Okay, we don't need it all to get to where we can do practical medicine. Okay, so we take what we need, right? And we leave the other stuff behind. You do the same thing in anatomy, physiology. You take what you need and push the others behind. I don't need to know all the names of the muscles. So I learned them long enough to take a test, and I forgot them. Okay, because I don't need that. If you were in physical therapy or like an athletic sports medicine, you would need those names of the muscles so you could impress your clients. That's really the only way they do. But they need that, right? Radiology, you need to know all the bones. I don't need to know all the bones, so I learned them at one point in time because to pass a test, then I forgot what I don't need to know, right? Or I put them in cold storage because the brain doesn't actually forget things. Okay, so this is one of those things that, again, sort of adds another layer to it. these neurotransmitters here that starts that signal, would that be a good way to say it?
Yeah, it attaches, like there's like sensors at the end of the dendrites, and so it attaches there, and then it sends a signal to the cell, and then the cell sends it off.
Yeah. So these epinephrine is a norepinephrine is a neurotransmitter. We learned about epinephrine, right? Sympathetic nervous system or norepinephrine. Don't really want to break down the differences, if any. Okay. So norepinephrine is a neurotransmitter. Okay, it's just another type. Okay, so norepinephrine. I'm trying to really remember, but I can't remember the differences. But norepinephrine. Okay, so you have that neurotransmitter. You have dopamine, which is a neurotransmitter. It's going to transmit something differently. Okay, a different signal. Okay, you have acetylcholine, which is going to transmit something different. Okay, and all these are just different neurotransmitters that will attach there and send a certain signal. Serotonin is a neurotransmitter. You know, people that have ADHD, they have decreased levels of serotonin in their brain. They don't have those neurotransmitters. So this is grasping, this is sort of buffering, but I think also they don't have these as many dopaminergic neurotransmitters, so they lack these neurotransmitters. They say people with depression, clinical depression, has a decrease of serotonin, okay, because they're responsible for transmitting these signals. Okay? So that's all just a different pot of goo, that we don't necessarily need right now. But I do want you to make sure that you understand that we're, at the beginning of class, like the beginning of the school year, I said we're just scratching the surface. We're just sort of riding along the surface and scratching the surface. We're still sort of doing that. Because we sort of dipped our foot in it and go, woohoo, that water's cold, I don't want to go there right now, let that warm up. But we're still sort of on the surface of it. So a lot more learning. This is why medicine is a continuous, lifelong learning adventure. Okay? Because you're always going to have continuing education. You're always going to have something else to learn. It's like the beautiful game of golf. Okay? You can't be good enough at golf, can you? You can't be good enough. Otherwise you'd win every tournament, right? And you eventually get bored at it. But if you follow golf, which you should, I don't know why you shouldn't, but if you follow golf, those guys are always working with their coaches. They have swing coaches, putting coaches. They have sports psychology that goes in and gets their mind in the right mindset. They have physio. People give massage and stretches them out, right? They're always working to improve their game because they can never get good enough at it. Now they may go for a period of time where they're winning. Ask Tiger Woods, right? They're winning, but at some point in time, something takes place. Their putting goes away. Their driving goes away. So they have to go back and work on that driving, work on their putting, work on their shots, work on their mental focus, right? All this stuff, they continue to work. Otherwise, they would win everything that they sign up for, right? So the medicine school is the same way. You continuously have to work on your mental focus, study, go back, study, pick up. It's very much like the great sport of golf. That's why it never stops, right? You never stop learning. Everybody good?
I have one more thing to add. Could you go back to the glycolysis where it showed like that messy stuff?
Am I going the right way?
Yeah. So before I even because eventually you'll have to memorize all of this, because the red stuff is enzymes and this is intermediates and all that. Before I even got to this, you still have to take general biology 1 and 2, chemistry 1 and 2, and then you get to biochemistry. So don't feel like all of this is going to be thrown at you in one class. This was just a general overview, but yeah, that's all I wanted to say, is that it builds on as you go through school. But yeah, I mean, it was in a way the surface level, but again, don't feel everything in there you have to know it very specific. I just added it on there so you know that it's a lot more complex than the general message that I was trying to give with this. But yeah.
That brings up a good point, because you learned general bio. You learned some of this in general bio, right?
Mm-hmm.
When do you take general bio?
Freshman year.
Freshman. When do you take biochem?
I took it my 3rd year.
3rd year. Did you need that information from general bio for biochem?
Because in general bio, we just knew that glycolysis creates 2-pyruvate. That's all we knew. And we just knew all of this was involved, but we didn't have to memorize it. But then once we got done with OChem and general chemistry, then we had to memorize every single little step there. There's glycolysis and gluconeogenesis, which is this but reversed, and then adding more stuff in. But yeah, that's not until your 3rd year, but it all comes back and haunts you.
So it's not learn and forget.
No, yeah, it's learn and retain. And then for the MCAT, after you graduate, you have to know all this.
Yeah.
I was getting PTSD.
Yeah, really? Yeah, that's true. So this 9 hours is not, you can see that this is not going to be very much here, right? You can sort of pick that up, that hey, my study time is going to have to really be increase. I'm not saying it's an impossibility, because it is possible. You go to medical school, right? So it's a possibility. But when we talk
Teachers,
about
pardon this interruption, at
study
this
time,
would you please release the gents to Mr. Nash and F104? Thank you.
This changes. Your study time changes because of the fact that when things get harder. But if you pick up these habits now, they won't be so hard to carry forward.
And it feels so nice once you learn it and you're able to teach it, because in a way feels rewarding, all those years of college. I'm able to teach this. It's kind of cool. It feels rewarding afterwards, to the point where if you go back and look at that information, you only need 5 minutes to oh, okay, yeah, I remember that. So yeah, I mean, you really have to keep on studying the material so that it doesn't leave your head. But
Questions?
I actually have the answer for a question you asked, because I didn't really explain it correctly with the facilitated diffusion.
Uh-huh.
So there's the sodium-potassium channels, right? And so the reason why they're different is because sodium and potassium are too large to diffuse through the cell, and so we need specific channels that are a specific size for sodium and for potassium to go through. So the potassium channels has a larger space in between, so that potassium can go through, and the sodium channels, and sodium's a lot smaller, it has a smaller space, because these two can't diffuse through the cell without, it's way too large to go through the cell. So that's an example of the facilitated diffusion, or facilitated transport.
Super. Good? Everybody good?